Good morning, everyone. Thank you for joining us again for this repeat of a webinar that NCSD first did in August entitled Point of Care Testing, Where Are We Going? And this webinar in August was co-supported by the American STD Association, or ASTDA. We had a technical problem back in August, so we are doing a re-recording of the webinar with our presenter, Dr. Charlotte Gaydos. And again, Charlotte, thank you for your time and for the ability to redo the recording of this webinar. As I said, the original webinar was co-supported by ASTDA and NCSD, and that webinar back in August did include welcome remarks by Bill Smith, NCSD's Executive Director, and by Dr. Case Rittmeyer, who is the current president of ASTDA. But as this is a repeat webinar, we'll be jumping straight into the content with Dr. Charlotte Gaydos. And Dr. Charlotte Gaydos is currently a professor in the Division of Infectious Diseases, Department of Medicine of the Johns Hopkins University School of Medicine, and has joint appointments in the Emergency Medicine Department and Epidemiology and Population, Family, and Reproductive Health at the Bloomberg Johns Hopkins School of Public Health. She is also a recent recipient of an NIH-funded center grant to develop point-of-care tests for STDs, so she is the perfect person to be doing our webinar this morning. And with that, Dr. Gaydos, please go ahead and begin.
Thank you very much, and thank you for inviting me to present this webinar today. So, where are we going? Well, if we look at STDs in the world, we have 350 million prevalent cases of STIs that are considered to be curable. 100 million chlamydia, 36 million gonorrhea, 187 trichomonas, etc. If we include the viral STDs, many of which cannot be treated, that brings us up to about 3 billion STDs in the world today. However, if we look in the United States, we can see on the left triangle that the prevalence of STIs in the United States is estimated to be about 110 million. However, this is a little different from new infections, or what we call incident infections every year. And if we look at what the estimated new infections are every year in the United States, it's about 19 million. And you can see they're relatively the same. Many human papillomaviruses are the most common, and then we work our way up through chlamydia, trichomonas, gonorrhea. Interestingly enough, HBV is the lowest number of new infections for incidence, but for prevalence, syphilis is the lowest number of infections that we see for prevalent diseases. So today our objectives are going to be to identify some of the current options available in point-of-care testing, and then to discuss the accuracy of both the old and the new point-of-care tests for STIs, and then to talk a little bit about the impact of point-of-care testing on clinical practice and patient outcomes. So the organisms that we're going to talk about today include chlamydia, Neisseria, trichomonas, Mycoplasma genitalium, syphilis, and herpes simplex. We won't have time to talk about HIV today, or HPV. So let's talk a little bit about what a point-of-care test is compared to a laboratory test. We're all used to sending our assays to the laboratory. It can take 2 to 4 days turnaround time before we get the answer. These assays are usually rated by the FDA to be highly complex or moderately complex. This means that they require a trained laboratorian with different degrees of educational training to perform the assays. Point-of-care tests can be CLIA waived, meaning that this is a lower level of complexity by the FDA, and these assays can be performed with minimal training and do not require a laboratorian to perform them. The nice thing about a point-of-care test is that they can take up to 20 minutes or an hour to perform, and so the answers are available to the clinicians fairly quickly rather than in a week or a couple of days. So one of the advantages of being able to use point-of-care tests in STIs in real clinical settings and not have to wait for the lab test to come back is that you can, of course, immediately treat the patient before they leave the clinic. This avoids loss to follow-up. Many people don't come back to STI clinics, for example, to get their results. So you've got them, you don't lose them, you can treat them. But we don't too often think about the impact on the actual epidemic of the disease. If you can treat the person before they leave the clinic, then you will decrease the interval of the disease spread. If they come back in a week, they've already had a week to spread the infection to their partners. So this is a distinct advantage to have an impact on the epidemic. And then also having a person know that they're infected and then giving them their treatment provides a unique opportunity to give counseling on risk reduction and how to practice safer sex. So it does provide an immediate feedback for a counseling opportunity. So let's talk a little bit about when is a point-of-care test good enough for us to consider using. Well, the WHO a few years ago has said that these point-of-care tests, at least for developing nations, and I think it's very applicable to the United States as well, should be affordable, sensitive, and specific, user-friendly. That means it's pretty simple to perform, rapid and robust, so that you can treat before the patient leaves the clinic, equipment-free, or at least minimally complex equipment, and then delivered. So we have the assured criteria that we have as our mantra to try to follow when these assays are being developed. Now, we all know that there's some older tests for STIs out there on the market. They're not very good. For chlamydia, we have the old Clearview, Biostar, the Quidel. They're not very sensitive. Gonorrhea, really, we just have the gram stain. That doesn't work very well for a clinician. Trichomonas, we can do wet prep. Not many doctors have microscopes in their office today. We do have the RPR and the VDRL, which can be done as point-of-care tests, but they're often not if they have to be sent to a laboratory. And for herpes, we have the old Zank smear, which nobody uses anymore, and it can't differentiate anyway between the different kinds of herpes viruses. So this is a cartoon showing some of the new tests that we're going to talk about today for chlamydia, gonorrhea, trichomonas, syphilis, and herpes simplex. Little pictures of what's coming down the pike. Next, first, before we talk about the actual tests, I want to spend a few minutes talking about what the clinician wants. We don't want manufacturers to invent a test that the clinician's not going to use because it doesn't have the characteristics that they need. So we have spent some time doing needs assessment for clinicians, asking them, what is it that you want in a point-of-care test? If you could design your own test, how sensitive does it have to be? How fast? How much? What about equipment, use of equipment? So we published a few of these studies, and the first thing we asked them is, what organism do you want to test for? And overwhelmingly, in various surveys that we've done and expert opinions and focus groups, 62% have said chlamydia, 14% have said an early seroconversion test for acutes for HIV, and then 8% syphilis. Overall, when you ask the participants their choices, which were significant, they wanted a sensitivity of at least 90%, a cost of about $20, a specificity of 99%, and a time of 5 minutes, if they could have their wish list. So when we used forced questions, or forced choice questions, saying, would you rather have this test or this test, with 16 13 simple questions, we were able to get a lot of information and put all of the data into a multivariate analysis model. And you can see under the first column of the red ring that says all, and these are expressed because this is a multivariate analysis as odds ratios for which things were most important. The most important for all of the tests, no matter which individual test someone chose, was sensitivity, and cost was second. This was the same if you restricted the analysis to the people who chose chlamydia. And there were 136 in this particular survey. The highest odds ratio, again, was sensitivity and cost, followed by specificity and the time, which are important, but were not considered to be as important for chlamydia. However, when you look at early HIV seroconversion and the people who chose syphilis, sensitivity was the most important, followed by specificity. So just an interesting way to look at what rises to the top for a needs assessment for clinicians. So we also then learned from this analysis, quite to our surprise, that cost was very important to the clinicians. So we did perform a cost analysis as part of a comparative effectiveness research model, and we modeled a new potentially chlamydia, gonorrhea, NAT test that was point of care that could return the results rapidly for patients. And so we found in the overall analysis that in the best case scenario, if there were a new potential point-of-care test that cost $33 or less, that had a sensitivity of at least 92%, and if women would wait 40 minutes to get their results, the results would be that 14 cases of PID would be averted by using the point-of-care test. So with the point-of-care test, because many people don't come back, and this all went into the model, I don't have time to go into the model in great detail, but the reference is there. But if you use the point-of-care test, there would be 66 cases of sequelae, i.e. PID, whereas with the NAT test in a clinic, there would be 80 cases. So you would actually prevent more PID, and you would save more money. Overall, the model was applied to a hypothetical cohort of 10,000 women, and this would save, under these assumptions of the cost and the sensitivity and the time, would save $70,000 or about $5,000 per case of PID prevented. So something to think about. Well, what about, that was all about the doctor or the clinician. What about the patient? Well, we know that willingness to wait is important. Willingness to be able to maybe collect a self-collected vaginal sample or a urine is important. And then if we ever do go to over-the-counter, willingness to pay will be important to patients. So this was an animated slide, but basically it reports a survey that we did with patients and asking the patients exactly what they wanted. And we looked at 5 different focus groups in Baltimore and Cincinnati, and all of the focus groups said they wanted something that was rapid, easy to read, simple to use, and they liked home-based testing. There were some barriers, but Hispanic women had a little bit different answers than some of the Caucasian and African American men. But this is a table from this publication, and basically they thought it was a great advantage to be able to have a point-of-care test done either in the clinic or at home. And you can see they said it's convenient, confidential, private, don't need an appointment, there's less embarrassment, and they could also serve to increase their sexual health awareness, and they felt empowered. So, and another actual questionnaire that we gave to patients in an STI clinic, we asked them which kind of specimen they would like to collect or have collected, and ask them questions about if they could buy it over the counter, how much would they pay, and how long would they wait. some of these things were important in the previous CER model that we did. So you can see that women do prefer to collect the vaginal sample or the urine sample rather than to have a cervical sample done. They didn't want to pay more than $20, which is about the cost of a pregnancy test these days, I think. But the key point here was, for us, was that 59% only wanted to wait 20 minutes and 20% would wait as long as 40 minutes. So you can see there aren't many that would wait longer or as long as an hour or even longer. We also asked them in another study, and this was done with women who had had a cervical, had collected a urine, and had collected a vaginal swab, and we said, how easy or hard was it to collect that vaginal swab? And you can see only 3% said it was hard, 80% said it was easy, and 16% said it was okay. So we have the idea that women will collect their own vaginal sample, which is good. Okay, just to, by way of background, not point of care, but we do know that now we have 5 companies that make commercial nucleic acid amplification tests for chlamydia. Many of them are second generation or greatly improved over the first generation assays that started in the 90s. Now many of them have moved on to expensive robotic platforms, so this limits their use in small labs and certainly in a doctor's office and also in resource-constrained settings. But just to remind everyone that we do have the ability to have very high sensitivities and specificities, and all of these assays are also approved for specimens that can be collected by the patient. We've even done some research with men to see how sensitive the assays are for penile swabs, self-collected penile swabs. This is just a picture of some of these large robotic platforms that you might see in large clinical laboratories or reference labs. And this is just a chart showing that you can see, it doesn't matter what the assay is, that there are very high sensitivities and very high specificities for both chlamydia and gonorrhea. Okay, so now it's time to talk about what's new for these point-of-care tests. So the first one I want to talk about is the GenExpert, and this is really not billed by the company as a point-of-care test, but rather a near-patient PCR test. And it does take 90 minutes, and so this is a barrier because we know that women might not wait 90 minutes. One way to get around this would be to change clinic flow so that the assay were started when somebody walked in the door rather than after they had visited the doctor and they're ready to think about leaving. So this could be what we would term a near-patient assay. This is the little cartridge here shown in this picture and the little machine. It's a very simple test to perform. It was FDA cleared December of 2012, I believe, and I'm just going to show you some of the results of the FDA clinical trial, which sampled over 1,700 females and over 1,300 male subjects. The nice thing about this assay is that it has a sample adequacy control, which shows you that human DNA is present, which precludes someone from doing an air swab instead of a vagina swab, or submitting water instead of urine if they were collecting the sample themselves. And also there is a processing control which controls for the lack of inhibitors that we sometimes see in amplified tests. So all good things. And you can see here on this slide that it doesn't matter whether it's cervical, vaginal, or urine, or whether it's chlamydia or gonorrhea, or whether it's male or female, that the sensitivities are almost perfect with very high specificity. So this is an excellent test as far as sensitivity and specificity goes. It's only cleared for male urine. That's what CDC recommends now for men for screening for chlamydia and gonorrhea. So this compares some of the older tests that I mentioned earlier, the Clearview, the BioStar. When you look at how they compare to amplified tests, the sensitivities of these older tests drops dramatically. Originally, they were cleared for the FDA because they were compared to culture. We know culture is not a gold standard anymore when you compare it to amplified tests. So these numbers, like the Clearview for cervical samples, 49% for the cervical, 32% when it's actually compared not to culture but to PCR, for example. So you can see here the expert assays stack up very well compared to some of these older tests. So I want to spend a couple of minutes talking about some new CT/NG tests that are coming along down the pipeline. There are many of them, maybe 25 companies or so working on them. I've chosen two to talk to you about today, but stay tuned because we're sure to have lots of new ones in the next 5 years maybe, trying to get through clinical trials and through the FDA. Neither of these are FDA cleared. There's two that I know more about, so I'm using them as an example. So the MA-MEF one first is one that we're working out with our collaborators at the UMBC, the University of Maryland, Baltimore campus, and it's based on MA-MEF, which is microwave accelerated metal enhanced fluorescence. It's based on detection of DNA, which there's an anchor probe which is anchored to a glass slide. The organism is laced with microwave radiation, and then the target sequence is from the sample is detected by a fluorescent probe. And this was published last summer, a evaluation of it, looking at chlamydia and actually just chlamydia in this publication, looking at it compared to amplified testing. And two targets were used, the plasmid and RNA, but the plasmid was the best, giving a sensitivity of 82%. It needs to be improved a bit, but it is rapid and it's cheap, and the reader that would read the results was only about $2,000. We're working on one for gonorrhea. This was presented at the CDC conference in June in Atlanta. So the way the investigators envision this is that eventually both the extraction of the DNA will be done through triangles on a slide that shrinkable plastic would push the sample through, and it would go through a filtering system, and then the MA-MEF assay would be run. So it's coming along. Secondly, I've chosen to talk about the Atlas system. This is a pretty small footprint. It is a PCR. All of the reagents are on this little card. Can go right into this very small reader. And it's simple to use, designed to eventually meet CLIA waiver. They're doing chlamydia this fall for CE mark in Europe, and hopefully chlamydia and gonorrhea in about a year in the United States. So here's a closer picture of the card. Everything takes place right on the card. This is an electrochemical detection with the loss of an electron changing ferric ion to a ferrous, or vice versa, I'm not sure which. Anyway, but you can see that you can get an electrochemical readout of a positive reaction here. And this has been published too, as far as looking at it compared to real patient samples. And the reference there is on the bottom, but has really good sensitivity and specificity when compared to amplified testing. So that's all I have for chlamydia and gonorrhea today, but stay tuned. We're sure to see some new assays coming soon. Well, now I'd like to switch to Trichomonas, and we all know about the wet prep. Yes, that's a point-of-care test, but it's not used very often. It's not a very good test. We don't often do electron microscope pictures of trichomonas on epithelial cells, but I included this because I think it's a really pretty picture of the trichomonas there, the upper right-hand corner, just glommed and stuck right onto that epithelial cell. So we do have two other point-of-care tests, the Affirm and the Awesome test, and we have research-amplified tests, which are not point-of-care, and these have two companies that are FDA cleared, the Aptima and the TVQ. So putting all these together, you can see that the point-of-care tests are shown in green in the middle there, that the Awesome test is about 83% sensitive. And you can see it stacks up kind of intermediate to wet prep and culture, but it's not as good yet as an amplified test. But nonetheless, doesn't require any equipment, and it's inexpensive, and it works very well. So here's a comparator between the Affirm test and the Awesome test. A recent publication by Cartwright in JCM showed that the Affirm, the Trichomonas part of the Affirm test, which also tests for yeast and BV, was only about 46% sensitive when compared to an amplified test, whereas the Awesome test in various publications ranges anywhere from 83 to 90. This is a picture of the Awesome test for those of you who have not seen it. It's pretty cheap, about $10, and you only get chlamydia or trichomonas here. You don't get BV or Candida like you get on the Affirm test, but it's a nice test. It's pretty much a dipstick and requires no training, no equipment. This is what the results look like. You get a red line which shows that the test works, and if you get a blue line, then this is taken to be a positive test. And this is based on mouse antibodies. It's a capillary bead reaction, and it looks for trichomonas membrane proteins. So the interesting thing about this is that Jill Hubbard and our collaborators have published on allowing adolescents to do this point-of-care test for trichomonas themselves. And you can see here that 38 times compared to the clinician test, they got a positive. And they agreed with the clinician 162 times, and they only had a very few that were discordant. And actually, some of those that were discordant that the self-testers, the adolescents got, were really, you can see, were positive. Of the 4 that the adolescents got positive and the clinician missed, 2 of those were real positive when you did a tiebreaker test, and all 5 of the clinician tests that the young adolescent girl missed were confirmed as positive. But it's intriguing. But why am I presenting so much on trichomonas? It's kind of a new kid on the block. We've ignored it for years, been focused on chlamydia and gonorrhea. I just want to spend a couple of slides showing you that TV is becoming very important in relation to acquisition of HIV. This is a graph from Patty Kissinger's article in the STI April issue, showing that of 2013, showing that of 14 published studies, 11 of them showed a increased acquisition over 1 for an odds ratio of acquisition of HIV. Many of these earlier studies were done in Africa, where there's a lot of HIV to be able to measure, but we're now seeing studies that are done in this country too. So I think we'll be seeing a lot more for trichomonas in years to come, and hopefully more point-of-care tests so that they can be used in clinics. This is just a report of several articles that have been done. The first bullet is that 2 investigators have estimated that the number of HIV transmissions in the United States due to trichomonas as a cofactor could increase the risk of acquiring HIV 2 to 5-fold. In another study, where we had 557 women who were not receiving any HIV ART therapy that were followed. They were cured of their trichomonas, and their genital viral load for HIV decreased by half of a log. And this was published by Anderson in 2012. So the genital load decreased, but the serum load did not for HIV. So then we have this model that's done by Chesson from CDC, using a mathematical model to estimate the probability of women acquiring HIV. And you could see that they think that these could be as many as over 700 new cases of HIV every year, which are directly attributable to the woman having trichomonas. And this would stack up for a cost of about $167 million. And they recommended that cost-effectiveness studies were needed. And lo and behold, this year we have just recently published this model on the cost-effectiveness analysis of trichomonas screening to prevent HIV. And the bottom line is that if we were following CDC guidelines and screening all HIV-positive women for TRIC, that we could save approximately $159 million. So important things to think about, I think. And I like this recent article that looked at trichomonas and bacterial vaginosis that we're not talking about today, but interesting, you can see that for these women who were studied and looked at viral shedding of HIV, there were 373, that if the woman had trichomonas alone, the odds ratio was 4. If they had trichomonas and BV, odds ratio was 5. If they had BV but not TRIC. But if they had both BV and TRIC, the odds ratio of actually increasing vaginal shedding for HIV was 18. So stay tuned. I think trichomonas will become an important player in our STI field in the next few years. So now we're going to switch to syphilis. We all know that the reverse algorithm testing has been introduced, caused a lot of confusion, but because of having point-of-care tests, we have seen that we've had a whole group of new serological tests that have been developed. And so having these tests is going to be very important in programs to eliminate congenital syphilis in the world, and also in MSM, both in the U.S. and the world with MSM who have HIV. So just by way of review, we know that we have the non-treponemal tests that we're all familiar with for years, the RPR and the VDRL. And then these new treponemal tests, some of them are pretty old, the FTA and the TPPA, but the new ones are mostly point of care. So neither of these tests are perfect, and they do have biological false positives, especially in the non-treponemal tests. And you can get cross-reacting treponemal test antibodies. And important thing to remember is that the treponemal tests, FTA, the TPPA, the TPHA, and some of the new point-of-care tests that we're going to talk about cannot distinguish active from inactive infection for syphilis. So if you get a positive treponemal test, you don't know if it's active infection or whether it's old treated or old resolved syphilis. So the new point-of-care tests, as well as the enzyme immunoassay tests that proliferated using the treponemal antigens, these are because we had the ability to make recombinant antigens. And so we had laboratories come out with changing the way they did syphilis testing from the RPR to doing an EIA or a CIA or an MFA to be able to look for the treponemal antibody first. So this has led to a lot of confusion and finally resolution by the CDC in that they have changed. On the left you can see the traditional way we used to diagnose syphilis, the RPR first, and then if it was negative you were done. and then if it were positive, then you confirmed it with a treponemal test, either the TPPA or the FTA or one of the treponemal tests. Now, these EIA and chemoluminescent assays are very quick and much cheaper, easier to perform, and easier for laboratories anyway to perform 100 at a time instead of doing 5 at a time for the RPR. So the recommended reverse sequence now is to do the treponemal test first, and if it's negative, you're done. If it's positive, then to confirm with the second test by the RPR, which will also allow, if it's positive, to tell you, yes, that it's syphilis, it's confirmed, and that it has to be, the RPR has to be tighter then to be able to follow treatment. So if the EIA or the treponemal antibody is positive and then the RPR is negative, then this presents a conundrum because you have discordant samples. And so the recommendation is to go to the TPPA, which is a second treponemal test. And if it's negative, then syphilis is unlikely because you've got 2 negative tests now. If it's positive, then the treponemal test confirms the first treponemal test of the EIA. So you have 2 treponemal tests that are positive and the RPR negative. So people are still confused and still trying to figure all this out, but it's pretty clear if you go down the algorithm tree. So here's just a list of names of all of the chemoluminescence and the enzyme immunoassays. There's a lot of them out there. But now we've had many of these treponemal tests be produced as immunochromatographic test strips, and they are point of care. The only one that's cleared in the United States is the Syphilis Health Check, and I think that the ChemBio, the dual path platform, which is a syphilis screen and confirm, or an SSCA, is either approved or about to be approved. So I want to talk a little bit about these point of care tests. This is a cartoon showing the ChemBio test, where you can actually have a treponemal test and a non-treponemal test, so you can kill 2 birds with 1 cartridge, as it were. So you can actually have your treponemal and your non-treponemal test. And there was just a recent publication I was delighted to see, I think it was just like in August or July, of this DPP test, or what they call Screening and Confirms, SSCA. So you can see here in this slide that compared to the RPR in this study of good many samples, about 250 samples, that the sensitivity was 98% and the specificity was 94%. So this is comparing this screen and confirm test to the RPR, and this next cartoon shows that it's compared to the TPHA and to the FTA. Again, sensitivity in the high 90s and specificities looking very good. So stay tuned, because I think this is a point-of-care test that we will get a lot of use out of in clinics and in resource-poor settings in the developing world. There's a SD BioLine syphilis test, which again uses these recombinant antigens from the treponemal recombinant antigens, and the nice thing about the next couple of tests I'm going to talk about is that they're excellent for the developing world because they can be stored at room temperature and they have a long shelf life. So this is just what the BioLine test looks like. You can read it in somewhere between 5 and 20 minutes. Alair has a syphilis TP test too, and it can be done on plasma or serum or whole blood. Again, you don't need any water or power or machines or platforms, and storage conditions are around 2 to 30 degrees room temperature. So here's how they stack up, and you can see Abbott has one, Omega has one, Qualpro has one, and Standard BioLine is. And then I talked about the SD and the Trinity health check. So this just shows that they all have pretty good sensitivity. The Abbott in some studies and the Qualpro have been a little lower than what we would like. But anyway, this is just showing a comparison test here. And so something that's new in point-of-care testing is, what if we can combine syphilis and HIV in the same test? This would have great advantages for a prenatal clinic, for example. So just going to give you a little preview of a couple of tests that are coming down the pike, to stay tuned. This is the ChemBio company again, and you can see this is an assay that looks for HIV and syphilis. It detects HIV types 1 and 2, and then the treponemal test for syphilis. So it seems to be an interesting platform to keep an eye on. Here's another one by MedMira called Multiplo. It only takes 3 minutes and has a very long shelf life, room temperature, no refrigerator, again, ideal for developing countries. And this is the STD BioLine syphilis and HIV test. So we talked about the syphilis test, but this is for HIV and syphilis. It has a long shelf life, room temperature, simple to do, read it in about 20 minutes. So let's switch organisms now, and the last one we're going to cover today is to give you an overview of herpes. We're not going to give you herpes, we're going to give you an overview of herpes assays. So normally, we use serology and look for antibody to diagnose herpes. We have the old Zank smear where we could actually take an ulcer and do a test and look for inclusions. You can culture the virus if you want to actually look at the lesions. You can type it with the Elvis test to see if it's HIV 1 or 2. But we do have a new amplified test that's FDA cleared for the virus for lesions for HSV 1 and 2. Shown in the little picture there, and it is an amplified test, though, and has to be done in a laboratory. But I want to mention that there's now an FDA-cleared point-of-care test for looking for the virus in both genital and oral swabs, and that's the so-called ISOAMP test. This is an isothermal amplification. Before we go into that, I'll just show that of the serology tests shown here, we have the HerbSelect and the Kalon. Kalon is not licensed in this country, but the HerbSelect is. And this is often used when we want to see if someone has had herpes or has herpes to look at blood. But there are some lab-developed tests looking at the lesions for the virus, amplified tests, not point-of-care. And then I mentioned the Q to the X PCR, which was FDA-cleared PCR. But the one I want that's shown in the red circle is the point-of-care test, and you can see it stacks up very well to the sensitivity of lab-developed tests and to the Q to the X PCR test that's FDA cleared. This is FDA cleared, showing a sensitivity of 97% and a specificity of 93. And so I want to just talk about that point-of-care test a little bit. Let's see here, next slide. Yeah. So this is FDA cleared, and as I mentioned, it's ISOAMP. It's an isothermal assay, and it's called a helicase-dependent amplification, an HAD. So you have more initials to add to PCR, SDA, and TMA. Now you have HAD, but it is an amplification, and it does give results in an hour and a half. So there were 5 study sites in the U.S. that were used for the FDA trial with an overall prevalence of 37%, and compared with the Elvis typing test, 98%. So the next slide shows a publication of the data that was used for the clinical trial, I believe. So the analytical sensitivity for HSV1 was 5 copies of the virus, and for HSV2, 34 copies. And for the FDA clearance, viral culture was used as the reference standard, and clinical sensitivity overall and specificity was 100% and 96%, respectively. So this is what the little assay looks like. When we say ISO-amp, it's not room temperature, it's 64 degrees, and it has to stay in this little heating block for 60 minutes. Basically, you have a swab in transport media, viral transport media, and you put 25 microliters in some buffer, put that in the ISO-amp machine there where the isothermal HAD happens, and then it comes out and it's put into this little point-of-care cartridge shown on the right. So it potentially makes it suitable for point-of-care testing, I think, an hour and a half. It's less than an hour and a half, so it's fairly fast. It doesn't meet our 20-minute requirement, but so here is a comparison between the Elvis HSV test, and you can see again high sensitivity and specificity, large numbers of reactions or tests that were done. So lastly, let's go to the new kid on the block, Mycoplasma genitalium. I think of this organism as similar to where we were 20 years ago with chlamydia. It is a sexually transmitted organism. It's very clearly associated with non-chlamydial, non-gonorrhea urethritis in men. Many studies showing it's associated with cervicitis in women. It's not so clear for PID and endometritis and adverse birth outcomes, but there are studies showing that sequelae are associated with Mycoplasma genitalium. It is the tiniest prokaryotic bacteria that we know of that's capable of self-replication, even though it doesn't have a cell wall. So it's very difficult to culture. There have been research PCRs that have been described that work very well. We don't have a FDA-cleared assay yet. The Aptima research use only TMA is the only potential commercial test that we have coming along the pipeline. We're hoping that it may become an ASR and analyte-specific reagent soon. Anyway, so it's not a point-of-care test, and we don't have a point-of-care test, but something to think about for the future, because I'm sure we will have a point-of-care test someday. These are a couple studies that we looked at just to see what the prevalence was in our STD clinic, and you can see compared to chlamydia, in females, the prevalence of Mycoplasma genitalium in vaginal swabs was about 24%. So it's a pretty common organism, and in males it was 15%. And so we looked in a multivariate analysis for both men and women. I've just shown the men here for the sake of time to show that in a model where we looked at Chlamydia, Trichomonas, and MGent, it was independent of gonorrhea, trick, and chlamydia associated with urethritis in men, and we had similar findings with the men. So I think that we'll see more assays for this organism coming, and hopefully some more point-of-care tests that will be coming down our pipeline in the future. It's of interest because it's been noted that doxycycline often fails to cure urethritis in men. It's recommended right now, I think, to go to azithromycin, which works a little better. However, Jørgen Jensen in Norway has discovered a single nucleotide mutation, much like in syphilis, that makes azithromycin resistant in treating Mycoplasma genitalium. In fact, a recent publication in CID showed that of all of the isolates, thousands of isolates that they've tested in Norway, 40% of them had this mutation and showed resistance to azithromycin, which then leaves us as the third line of treatment to go to moxifloxacin. And I just heard recently that they've discovered some moxifloxacin resistance. So these men that have these cases of urethritis that hang around for years and years, we may be getting into a situation like with gonorrhea with a STI that's untreatable. So anyway, something to keep your eye on. So let's end now with, my next to the last slide is we also like with point of care to think outside of the clinic or outside of the box with maybe going to the internet. We started an internet project in 2004 where individuals, particularly young people, could collect a sample at home and mail it in and get the results and then get sent to a clinic for treatment. We recently gave our website called I Want the Kit a facelift. It's only active in the state of Maryland and in D.C., but we've put in some new features such that there's a risk quiz that individuals can take to see if they're at risk of having an STI. They have to log on with a HIPAA-compliant password and a protected login, and then they have to select a clinic before they order their test. And we have new instructions for home collection and more educational material. The website automatically sends them a text or an email, whichever they choose, to tell them that the kit was mailed, it was received, and that their results are ready to be retrieved and to go online. So we're hoping to increase our use of this test by particularly adolescents who probably don't want their parents to know that they're sexually active. So for point-of-care tests, I think we conclude that we have a promising future where we can take advantage of self-collection, even self-testing. We have better NAT assays, so we have more tools in our toolbox. We can think about testing outside of a clinic. A lot of interest now in having some of these tests be done in pharmacies so that people don't have to go to see the clinician. We will have, because of market competition, cheaper test kits. But what we need to do is learn how to use research to remove some of the barriers to get people tested. So I guess we have a lot to learn to effectively use our new tools in our toolbox and use research so we can detect more STIs and increase the number of patients that get treated. And I'd like to acknowledge all the people that work in my lab and the HIV lab with us, particularly those that are named in this slide. So I'm happy to take any questions if we have that mechanism, but I don't think we do. If you have questions, I'd be happy to have an email from you to answer your specific questions. So that's all I have for today. Thanks very much for your attention.
Thank you, Dr. Gaydos. We really appreciate you redoing this recording so we can make sure that those who attended the webinar and those who weren't able to are able to get this great information that you shared. Maybe you could you share your email over the phone here so that folks on the recording could contact you? Sure.
It's CGAYDOS@JHMI.edu.
Great. Thank you so much. We really appreciate your time and look forward to working more with you on these issues.
My pleasure. Thank you very much.
Thank you very much.
Bye.
Bye-bye.